Okay, thanks, Emilio. I would like to first thank the committee to invite me to present Dr. Fan. It's an honor here. Let me introduce Dr. Fan a little bit. Dr. Xiaohui Fan is a Qiushi Distinguished Professor of College of Pharmaceutical Sciences at Zhejiang University, and he's also the founding director of the Innovation Center of Yangtze Delta at Zhejiang University, China. He holds a Bachelor of Engineering in Pharmaceutical Engineering and a PhD in Pharmaceutical Sciences from Zhejiang University. He also did his postdoctoral doctoral training at the
Because we have lost you, possibly. I mean, the microphone may... Okay, it's back now.
You hear me?
Yes.
Oh, I'm sorry. Um, well, anyway, he did a lot of great research. Thereafter, he was appointed as a faculty member at Zhejiang University. Well, his research interests includes developing system biology based computational and experimental approaches to advancing regulatory science for Chinese medicine. He has published a lot of papers, over 100, on top journals, and over 40 patents or software. He is a member of uh, a lot of scientific journals, editorial board, and he also has also been granted a lot of there are a lot a lot of great awards by the Chinese government. And he's also holding a lot of positions like deputy director of State Key Laboratory of Chinese Medicine Modernization, or and an honorary researcher of the University of Sydney, a visiting professor of FDA and a commission member of Chinese Pharmacopoeia, and invited external expert of Chinese NMPA, and the president of Massive Analysis of Quality Control Society. Well, anyway, so Dr. Fan is a very distinguished professor in China, and today he will share us a speech, a webinar basically, with the title of Transcriptomics-based TCM Network Pharmacology, Theory, Methodology, and Applications. Let's welcome Dr. Fan.
Okay, good evening, or probably good morning or good afternoon, my dear colleagues and friends. First of all, I'd like to thanks the invitation from the IUPHAR. I'm not sure, can you guys see my screen?
Yes.
Okay, great, thank you. Yeah. First of all, I also like to thank Li Qing for the nice introduction, and especially in the poor internet condition. As I mentioned, I'm Xiaohui Fan from College of Pharmaceutical Science at Zhejiang University. And actually, for me, it's a great honor to join the Pharmacology Education Initiative today. I'm excited to have this opportunity to share our research on this topic. Basically, the transcriptomics-based TCM nanopharmacology. Of course, the TCM stands for the traditional Chinese medicine. And here's the outline of my talk today. We will start by setting the stage of some background on complex disease and how our understanding has evolved. And then
I think this, yeah, the sound is off. Sorry, your sound is not
Excuse
working.
me?
I'm sorry.
Oh,
We
okay.
cannot
Sorry for
hear you.
that.
And
Let me try.
someone,
Uh,
I cannot
just
see
1
the
minute.
slide too.
Yeah, I'm going to switch to another internet. Let me try. Just 1 minute. So with that, can you still see my screen?
Yes, I can see the screen.
Okay, great. Thanks God. Anyway, here's the outline of presentation today. I'd just go through, after the mature transcriptomics, we're going to go through all the works in the transcriptomics-based network pharmacology. And at the last, we'll run up some conclusion and exciting future directions. Now here's the background. Actually, we all understand that there is a long way to understanding the disease, especially the complex one. So a lot of technology has been invented to deal with this complex situation. I just list a bunch of them, as you can see from here, especially since the last 4 or 3 decades. The first technology I'm going to mention is the DNA microarray. Although right now, probably we don't use it anymore, but it's absolutely is a very important milestone. And especially in the 2000, the citizen biology was approached, which set the tune for the research I'm going to introduce here today. And of course, after that, especially in the 2007, right, the network pharmacology was invented or introduced in the area. And after that, the single-cell RNA-seq network medicine, spatial transcriptomics, and so on and so forth. Most of the technology I'm going to mention in my topic today, and especially the single-cell RNA-seq and the spatial transcriptomics at the end of my topic. So let's start with what's the definition of the network pharmacology. And of course, I believe most of the audience here today are quite familiar with the network pharmacology. Basically, from my point of view, is provide a concept and tools decoding the complex system, and especially using the nonlinear research pattern. I just did a very quick survey in the PubMed. As you can see, the papers published in this area is increased very, very fast. And a lot of it actually published in our journal in the pharmacology research. And for the network pharmacology, there are also several other definitions. One of them, is my quite favorable one, is the approach to understand the drugs and encompass the system biology, network analysis, connective, and so on and so forth. And more importantly, actually, we believe that it offers a way let us to rethinking about the drug discovery, and by the simultaneously embark on the effort to both improve the clinical efficacy and understand the side effect or the toxicity. So here's the very briefly, the history of the how and when the nanopharmacology was proposed. Originally, it's proposed in 2004 by the Professor Barbosa. He proposed a concept called the network biology. And after 3 years, there's a pharmacologist from UK. He proposed, he got the first step and proposed the network pharmacology. And right now, I personally prefer call the end to the next level. I call it as like the network pharmacology 2.0. Okay, this slide is basically I want to visualize the concept by showing you guys the social network. I assume every one of you have this kind of either with the Facebook or the X, or in China we use WeChat. Whenever you have a lot of friends, right? So if you have the friends, you can using this kind of tool to visualize the friendship by using the social networks. And of course, you can hear that if there's a lot of the people's you have a lot of friends here, and the dot basically is very, very bright. So by this kind of visualizing help us to understand not just the individual components, but how everything is interconnected. And another, the analysis like here is a show the domestic flight information in the States. If anyone of you are quite familiar with the USA, I think it's again very easy to identify the harbors, the city. I mean, now here's for instance, here's Honolulu, and these are the big cities. The majority mega cities are in the California, and also there's a bunch of cities in the New York or Miami and wherever. So imagine that if we not visualizing the domestic flight information, and we're using the same approach to visualizing the genes of the human. So we also will find several of the genes actually is quite important, and we'll get a very the data is very big, right? So of course, is in the in biology we can using this kind of technology, especially the network biology, to help us to visualize the interaction between the genes, and eventually we can using this kind of technology to optimize the drug development and discovery. Okay, here comes how we actually do with the network pharmacology. So let's look at some key applications. For instance, we can build the drug target network. And excuse me, can you see the mouse? Probably I forgot it. Okay. So for instance, we can using this kind of technology to develop or build the drug target network. It's probably the most important or the most commonly used research in network pharmacology. Here is the way I just give you guys a very brief or simple ways how we can discover the relationship between the drugs and the targets, and how we eventually visualize them using the network biology approach. And first, we can screen or research in this kind of drug-related database, and like the drug bank and so on and so forth. And also, I also list several very important databases in the TCM research. And then on the other side, we can also discover or through the data database research, we can identify the disease-related this kind of genes or targets. And through the text mining, or either way using the in silico approach or the the omics approach, you eventually identify which target and which drugs have the connection or have this kind of relationship, and we connect them and visualize them and then analyze them. So using the network analysis approach, we can either way we can do the drug repositioning, or identify the side effects, or evaluate the efficacy, or decode decode the mode of action of these kind of drugs. Okay, here comes the TCM. Why a lot of research in China, especially in China, are using this kind of approach. I mean the network pharmacology to investigation in the area of TCM. I assume most of the audience are familiar with the TCM, or at least you guys heard about that, since you're all the readers or the authors of the pharmacology research.
Excuse me,
Of course,
Dr. Fan.
Okay.
The slides haven't been advancing. So we're
Okay.
still seeing slide number 6 with background, the complex social network.
Can you see the slides?
Yes.
Yes, yes.
Which page, Dr. Fan?
Yeah, I still didn't see your share screen. Start from the the number 9.
This one?
I can't see your screen.
Yeah, this is page 9.
It's page 9, but somehow I just
Yes.
cannot see.
Yes.
Why?
And this page 9.
Just 1 minute.
This one. It's okay. Just tell me which page. This I will.
Yeah, the problem
go
is
on.
I cannot
This
see
one?
your screen.
Can you see?
No.
No.
Well, Xiaohui, you probably can try play your the PPT slides on your computer, but not share it. Then you will just go ahead, read it. I mean, do your presentation. Tintin can probably try.
Yeah, I can go
look at-
on, and Dr. Fan will interpret the slides to us. Okay.
Yeah, but the problem is I just okay.
You just can't
That's
see
so
the
weird.
slides.
Yeah. Okay, okay. Yeah. Okay, for the slide 9. The background of TCM.
This one is the TCM network pharmacology, how to uncover the complex relations between TCM ingredients and
one page earlier. Flip back. Page 9.
page.
Yeah, page
Yeah,
9. Yeah, okay.
this one. Yeah, this one is page 9. Okay.
I'm really sorry for that. I just anyway, I just going to introduce the page 9. As we all realize that TCM is very important in China, and of course this kind of botanical drugs are also very important in many countries, especially in the European. I'd like to give one of the examples. We call it the Zongjing formula.
Yeah, this one, just this page. Yeah, it's right.
Oh, sorry. Yeah, Zongjing, the formula is actually probably the most famous formula of the TCM in China and Japan and Korea. So for those Zongjing formula, we probably have like hundreds of formulas, and in total they will have like 116 herbs and contain more than 5,000 compounds. So it's, as you can see from here, if we're using this kind of network biology approach to visualize their interactions or connections, it's quite complicated. So next slide.
Okay.
So for the TCM research, if we using this kind of approach, especially the nanopharmacology approach, basically we are going to uncover this kind of complex relationships between the TCM ingredients and the biological effects. And because there is a kind of a hypothesis behind that, the rationale is we believe that the efficacy of TCM is up to the complex interaction between the ingredients and this kind of whatever we call the biomolecules. Basically, the genes or protein or targets. So next slide. This picture I actually adopted from our recently invited review for the Chinese Journal of the Nature Medicine. And this picture, basically, we're going to show you guys how we can develop or establish this kind of ingredient target relationship through this kind of either with research database or using the in virtual omics kind of approach and so on and so forth. After that, as I mentioned earlier, we can either we develop this kind of ingredient targeted network or the disease gene network. We combine them together, we will get the ingredients and the gene or the proteins and then eventually the kind of target networks. By analyze this kind of network, we eventually we can investigate the mode of action or identify the active ingredients from the botanical drugs or TCMs. So next slide.
Okay.
So as I just mentioned, there are several ways to identify or discover the relationship between the ingredient targets. One of the commonly used ways is using the system biology approach, or called the omics approach, to link our TCM to these kind of targets. You can either way investigate using at the DNA level or the RNA level or protein or metabolite level. So there's a lot of all kinds of omics. I'm going to talking about today is a transcriptomic approach, basically at the RNA level. What I'm going to say is not the proteomics metabolites is not important. That's not true. Actually, in most cases they are very important. But one of the reasons why my group and a lot of researchers here in China and in the international in the international universities, they're also using the transcriptomics because this kind of technology already be developed for many years. We actually back to like several years ago, we summarized the in the transcriptomics their applications in traditional Chinese medicine. And basically, it's a very efficient tool and widely used tool in the traditional Chinese medicine studies. So next slide.
Okay.
However, before we going to dive in the transcriptomics, we must understand the complex of the transcriptomics. One thing is that if we're talking at the gene level, I'm not sure whether you can realize how many genes do we have in the human. This slide, sorry, Tinian, are we staying the same page? Is page number 14?
This one? The last slide?
14.
14.
The complex of the complex of transcriptomics.
Okay. This one, yeah, the page 14.
Okay, yeah, sorry, since I cannot see your screen somehow, I don't know why. So at the gene level, we are very, in most cases, we just only focus on the protein coding genes. They are roughly like 20,000 genes. But we also, nowadays, we also are very interesting about the non-coding genes. There are many of them. So in total, there's roughly like 78,000 in total at the gene level. If we're talking about the transcriptomes level, is the number is increased dramatically. So as you can see from these slides, this actually is pretty hard and very it's a challenging problem. We measured all the genes expressions in the same experiments. So next slide.
Okay, 15.
Yeah. So I'm going to briefly introduce for the past almost 20 years, my lab working together with the international collaborators working in the area. We try to answer one of the questions: are transcriptomic data reliable? Of course, it's very important for both the industry and academics. So started like 2004, and we set up this kind of consortium called the micro quality control. Eventually, we spend this kind of MAQC to the sequencing and the single cell sequencing and so on and so forth. With this kind of organization still working around now, and we already published several papers in the Nature series journals. The recent one is actually published like 2 or 3 years ago. And there is a collection in the Nature, in the homepage of the Nature collections. So I just go through the results, the brief results of the MAQC studies. This next slide.
Okay, 16.
Yeah. This slide basically is a very interesting story. Back to really 20 years ago, There's a paper published in Science. Basically, they using the very same pair of RNA samples using different, platforms. One is Affymetrix, another Angelin and Amarcia, and eventually you can find only 4 genes for differential pair genes are in common. So that's really raises a lot of questions in terms of the reliability of the transcriptome state. So after that, we set up this kind of MQC consortium to investigate the problem. If the transcriptome, this kind of approach is not that okay for the study, no way we can use in this kind of technology to do, especially in the nanopharmacology research area. Oh, next slide.
Okay.
So fortunately, after probably 2 or 3 years later, we published a set of papers in Nature Biotechnology. And the main take-home message here is the microarray technology, basically the gene expression data is reliable. And of course, only the SOP and the proper data analyze approach are followed. And here's the same data we analyze using the different analysis approach. As you can see, the common DGs dramatically increased from 4 to the basically the 39. So it increased like the 10 times. And this kind of research eventually resulting we published or eventually comparing with the FDA's guidance for industry in the pharmacology jumps data submission. And next slide.
Okay.
Of course, after that, we didn't stop it here. So we launched the MQC2. We try to investigate other genomics biomarker are also reliable. And in 2010, we published a bunch of papers that demonstrate, yes, of course. And the main the conclusion is, of course, the genomics data can provide some reliable information in terms of the this kind of a biomarker or the diagnosis or prognosis models. Next slide.
Okay.
And for the third phase of MQC, we investigate, try to answer are the NGS are reliable or not. And of course, at the very beginning, we just investigated the microarrays. But after 10 years later, most of the laboratory in the industry, they don't use the microarray anymore. They use all kinds of the NGS data. And then we also, here we published a bunch of papers, including the Nature Biotechnology and Nature Communications and so on and so forth. And basically, also here, the massive undertaking here, we have the community set up this kind of NGS as the golden standard for the genomics the research. Next slide.
Okay. Page 20.
No, 21.
Yeah, page 20.
21.
21.
Already, yeah, 21.
Okay. This one is 21.
So the most recent MQC research is focused on to understand the reliable or not in terms of the DNA sequence. For instance, here this paper will reported reliability or validation of the ctDNA, which of course are also very important for the precise oncology and precision medicine. So this MQC consortium is still ongoing research project, and so we are actually open course for anyone interested in this. If you guys are interested in this area, and we will be more than happy to welcome you guys to join this consortium and working together with us. So next slide.
22.
Yeah. So basically, after this kind of long journey in terms of the reliability of the transcriptomics, we believe that this kind of technology, I mean the transcriptomics, is much reliable than the proteomics or metabolomics. So that's the main reasons why using this kind, mainly uses this kind of technology to in the network pharmacology research. And the following slides, I'm going to take you guys go through all this kind of research in terms of the transcriptomics-based network pharmacology, mainly focusing on the efficacy, the mode of action, the drug design, and eventually I'd like to introduce several new technologies in this area. So next slide.
23.
Yeah. The first case I'm going to show you guys is I call it the recovery of the disease-induced imbalance network. Have you guys ever heard about the Chinese medicine or traditional Chinese medicine? The balance thing actually is the core concept of the TCM. So we believe actually for the normal peoples, there is a balanced network. And then once you get the disease or whatever or injured, there is the network became the imbalanced. However, after the treatment, especially using the TCM, there is a recovered network. So there's a whole concept. We're using the transcriptomics approach to develop a set of tools to quantitatively measure the recovery rate of the network. So here's how we develop and build this kind of imbalanced network modeling. Basically, of course, we cannot directly measure this kind of gene expression as the human. So most of the case, we're using the rat model, animal models. For the animal models, here's in this paper, we just using this kind of animal model, AMI model. We measure the differential pairs of genes and connect with the disease-related genes and using the PPI, the protein-protein interaction. So eventually we got this kind of imbalanced network. So here's the red dot basically means the upgrade gene, and the first is the balanced network. After the AMI red model, we measured the differential expressor genes. We measure which genes upgrade and which genes are downgraded. We can eventually build the imbalanced network. And then after the treatment, here's is a traditional Chinese medicine called the sema injection. Now it's quite famous in China. Basically, this kind of TCM is composed by two herbs. One of is the red ginseng. I assume probably some of you have heard about that. This is the kind of ginseng, also very popular in China. Another is called is the radix officinalis. You guys might not familiar with it. In China, we also call it maidong. So these two herbs, they made after the manufacturing, they made of this kind of TCM called sema. After treatment, you can see the most of the either the red one or the green one, they are recovered close to the balanced network. And of course, we further using this kind of animal model, we just using the echocardiography results to compare the TCM treated one, and also using only the herbs treated, one of the herbs treated. So as you can see here, there is a dot in the TCM, the sema injection here. Yeah. So which means actually means that the combination of the TCMs is much better than the single herbs alone. So it's a very interesting observation. So next slide.
Which
The second case, 27.
Oh, yes, right.
the second case I'm going to show you guys is using the nanopharmacology approach to investigate the traditional Chinese medicine serial. Here is the case we call is 君臣佐死. I know for the most of our audience that probably cannot understand it. Basically, is the herbs in one TCM they play in different roles. One of the herbs actually is the major roles. We call it like the Jun is a king or emperor, and the second one is a minister. Then there's also some like the assistant or and so on and so forth. So there's a kind of serial in the traditional Chinese medicine behind the drugs. So here, this study, we're using this kind of nanopharmacology approach to investigate whether there is this kind of 君臣佐死 in this kind of TCM in terms of the recovery network. Okay, here's the next slide, please.
Yes.
So here's a very brief introduction of the the drugs, the TCM drugs. I'm not going to mention the name. It's not important. But the one thing I just want to emphasize is this kind of TCM actually is composed by 4 herbs. The one herb is actually the upper left is the king, which is also called the dream. And the rest is, as I mentioned, as a minister or assistant, colonel, and so on and so forth. So next slide. So in this, we designed a transcriptomics study here. Basically, we're using the whole TCM, and we also divided this kind of TCM into 4 parts, each single herbs. And we extracted them and to treat the animal, and we did the transcriptomics assay. And we were trying to identify the which gene are differential priced, which protein differential priced. The next slide.
Okay.
As you guys can see from here, basically the whole formula, the original TCM has the best efficacy, right? It's of course not it's acceptable, right? As we expected. And also in terms of number of genes, differential pressed genes or the proteins, also they have the most regulated number of genes or proteins. So next slide.
Okay. 31.
Yeah, 31. Yeah. So we're using the same approach to develop this kind of the AMI induced imbalance network, and we measured the whole TCM and the 4 individuals here, and we calculate how many this in terms of the recovery rate of the imbalance network. So as you can see from here, the first one is as I mentioned, is he actually is a king, right? So plays the most important, the major role in terms of the recovery of the imbalance network. And of course, you can see here the number is the largest one. So it is quite interesting result here. So we also report out this kind of observation in a journal called Chinese Medicine. So next slide.
Okay.
In the third case, I'm just going to show you guys how we use this kind of nanopharmacology approach to discover the active ingredients from the botanical drugs, from the TCMs. Of course, we all know that the TCM have a lot of the ingredients, right? Thousands, hundreds, thousands of them. So the scenario behind that is we're trying to identify a bunch of all the group of the ingredients in terms of the recovery rate. is equivalent to the whole TCM or the original ones. So this is the hypothesis behind this kind of study. So how we did this kind of study? Let's go the next slide.
Okay.
Okay. For instance, in this case, we're using this. There's also another Chinese medicine called Xiaohui Fan. Just forget the name. Basically, it's a kind of a symposium from one of the very popular herbs in China. And we developed a method called the content-weighted network-based efficacy. We calculate, the targets. Basically, how many targets and how much the targets was basically the impact or influenced by this kind of components, and we combine them, and here's the results. At the end of the day, we find 5 of them, the combination is probably equal to the 96% in terms of the recovery rate of the whole network. Basically, we only use this kind of five components, 5 ingredients. We group them together. Is pretty much like equal to the original TCM in terms of the efficacy. So with next slide.
Okay, 34. Yes.
We can consider this as a kind of new combination or new drugs, whether it's equivalent to the original one in terms of the efficacy. As you can see from here, the combination of the 5 ingredients and significantly reduce the effect size in the animal model. So we conclude that this kind of 5 combination of ingredients are the active ingredients of this kind of original Xiaohui Fan. Okay, so here comes the 4th case. Next slide.
Yes. 35.
Yeah. Basically, this is the paper published by several colleagues from Henan University, not from my group. Their research paper actually basically demonstrated how to use this kind of nanopharmacology approach to discover the mode of action of TCMs. And here, this Chinese medicine, of course, also very famous in China, is a Qingfei Jiedu. If you ever heard about that, it's kind of to treat like the severe pneumonia in China. So of course, it's very popular, especially in the COVID. Next slide.
Okay.
And here's the workflow of how they, by using the integrated nanopharmacology, by integrating the network biology and the transcriptomics. Basically, they first developed a compound and target network, and they identified the co-targets associated with this kind of disease, and using the PPR network to identify the potential target genes. So here's the results. Next slide.
Okay, 37.
Yeah. So basically, there's the whole workflow is here. I just go through this. As I mentioned, there's a bunch of database. You can search the database, identify the bioactive ingredients, and here they set some criteria like the ADMET and so on and so forth, and also they search in terms of disease from the gene card, OMI, and drug bank. So got this kind of targets, they find the common targets and using to identify and develop this kind of ingredient and the disease network and perform the network analysis, as I just mentioned earlier. So next one.
Okay.
And they also validated their results. And just very quick go through here. And then they did a bunch of in vitro and animal study to validate whether the target they identified is correct or not. Okay, next slide.
Okay.
So starting from this slide, I'm going to present you guys several new technology. Is also very important in the transcriptomics-based nanopharmacology research. The first one is I'm going to introduce the single cell and the spatial transcriptomics. Next slide.
Okay. 40.
Yeah, 40. Yeah. So mentioned about the single cell and the sequencing, I assume most of the audience probably already heard a lot about that. Since we all have like the regarding the humans, right? We already have the like over 40 trillion cells. So the heterogeneity actually is very important in terms of the disease research. So unfortunately, go back to the old days, especially in the nanopharmacology research, especially we using the transcriptomic approach. We use actually in most cases we only use the bulk sequence, right? So we basically measured which genes upgrade, which genes they downgrade, but we didn't know these up or downgrade genes came from which cells. Since as I just mentioned, the heterogeneity actually is very important in the disease. So we for long days actually we actually hopeful there is a kind of technology can resolve this the kind of problem. So next slide.
Okay.
So fortunately, as I just started the very first slide, as I mentioned, since 2009, there's a new technology. Very powerful and very popular right now in the in the disease area, and of course, I just listed here that they be selected as the method of the year of the Nature methods and the breakthrough of the year by the Science Journal and so on and so forth. So which means, as you can see from here, this kind of technology got a lot of tremendous attentions from all the research area. But another thing is, although the single cell sequence is very powerful, but there still has a drawback, the major drawback of this kind of technology, because this kind of technology at the very beginning, the dissertation of the cells from the tissue sample, we lost actually the location, where the cells came from. So after like 6 or 7 years later, in 2016, there is a science paper published in Science. They proposed another technology called spatial transcriptomics. Of course, here is also, like 2 or 3 years ago, is named the method of the years by the Nature methods, and the most important technology by the Nature. So both two technology, of course, is very powerful, and we, of course, would like to incorporate them in the nanopharmacology research. Next slide.
Okay. I see 42.
Yeah. I'm not going to go too much detail in terms of the technology itself. There's many review papers in this area. If you are interested about it, we also published a cover story in China's Life Science, or recently, just a couple months ago, as a cover story. We have just reviewed the whole technology. But we are here is I'm going to present you guys one of the example. We're using the single cell transplant approach, how to incorporate the network pharmacology together to investigate the kind of botanical drugs. Here is one of the active compounds from Tensional. Basically, here's the compounds, and the name of compounds is Tensional IA, and in this kind of AMI models, and we're utilizing the single cell sequencing approach to sequence all the CD45 positive cells in the heart of the MI mice after intervention. This kind of compounds for 3 different time points. So eventually, we're using this kind of single cell data, as you can see from the left up, these kind of figures. As you can see over here, there's many cells. Our sequencing using the kind of technology, we can identify which subset of the cells most contribute to the recovery network of this kind of treatment. Next slide.
Okay.
Okay, here's basically how we utilize this kind of technology and the data. So I just quickly go through this here. This paper already published in Small Methods. Very interesting about this story. I probably you can read our papers. So next slide.
Okay, 44.
Yeah, another new technology, at least in my lab, we pay a lot of attention is we call it the cell-cell communication or interaction network model. We believe that's very important in terms of the system biology or the network analysis, and especially the concept. This concept is perfectly match with the Chinese medicine, the philosophy behind that. The very have share a lot of comments. So next slide.
Okay.
Basically, this slide is briefly introduced in case you're not familiar with the CCIC cell-cell communication or in right, the PD-1, PD-L1 is actually the main, very important ligand receptor between the two cells. So understanding the cell-cell communication is very helpful to understanding the disease and and also the investigation of the drug efficacy. Is kind of cell-cell communication. Mainly, they can be divided into 3 types. One way I call it the physical contact dependent communication. It's very easy to understand if 2 cells they are very close to each other in the tissue sample. Of course, they have more likely to have the communications, right? Another is we call it the chemical signal dependent communication. Including like the short range and also a lot of the long range. They mainly communication with each other through one of the cells. They send some ligand, and other cells have some receptors. And the last type of cell-cell communication actually is I call it actually is interesting. So one of the ways to inference this kind of cell-cell communication is using the single-cell data. Once you got the single-cell data, you can annotation the cell types, and either way from the chemical signal dependent point of view to discover this kind of cell-cell communication, or you can combine the location data. Here's particularly you can use in the spatial transcriptomics data, combine them together to identify which cells they not only close each other, also they have kind of this kind of communications. Next slide.
Okay. 48.
Yeah. Here I just show you guys several very popular software or the algorithm in this area. Widely used algorithm, including the cell chart. They published original published in the Nature Communications, and the 7DB. There's a database which basically they collect this, which is the ligand, which is the receptor, and which like receptor complex. And they are originally published in the Nature Protocols in 2020. Next slide.
Okay.
And of course, you can also inference this kind of sensor communication using the spatial transcriptomics data. There's also several quite popular this kind of algorithm here, like the GTO here. So I'm not going to show you guys too much details about the algorithm. Just if you guys, some of you are interested about the area, you can just take a photo of this and go back to read this paper. Next slide.
Okay.
So of course, in this area, our lab we also developed a bunch of tools to basically to identify, including the database and algorithm. In this slide, we showed an approach called assay crosstalk approach. By utilizing a database manually occurred in our lab, we call it CellTalk database. Basically, we use this kind of ligand and the receptor pairs in human and mice. And we identify which cells or which cell types have this kind of communication. And of course, we can further incorporate this kind of information in the network development and then in the network construction and then in the network analysis. So next slide.
Okay.
And of course, we can not only using the single cell data, we also can using the spatial transcriptomics data. In this paper, we published a method called SparTalk. We can construction this kind of network made by the spatially solved transcriptomics data among these kind of cell types. This paper was also highlighted by the Nature Communications. Okay, next slide.
Okay.
This slide briefly introduces the most recent papers in this area, as published in Genome Biology just a couple weeks ago. We developed an approach we call MyTalk. Using this kind of approach, we can inference the cell communication mediated by the EV derived, also the microRNA target interaction between the cells. So as you guys can see from this slide, we can not only develop or construct this kind of network using the gene interaction, we can also construct the network from the cell-cell combination point of view. Also, of course, how to define which cell have this kind of a connection. We can using the ligand and the receptor pair, or we can using in this slide we proposed using the microRNA and the RNA of the target RNA interaction. So next slide.
Okay, 53.
Yeah, and here comes probably the last 2 or 3 slides. In this slide, I try to show you guys some of the future challenge and directions in this area, especially in the AI-based area. In the AI in nowadays AI, everybody using all kinds of AI, right? So you using all kinds of the big models. So if we're using this kind of foundation models, I believe we can probably use advancing this kind of nanopharmacology approach with the AI, especially in terms of the cell communication network construction and then analyze. Okay, next slide.
Okay.
And here's the conclusions of my presentation here today. And also I'd like to propose some perspectives. The first take-home message will be the transcriptomics technology, I think is reliable if the SOP and the proper analysis I followed. So for the last almost 20 years, through the the investigation from the MAQC consortiums, we demonstrated that the transcriptomics technology is a very powerful and useful tools for both the nanopharmacology research and of course they can be used in many other areas. And if we use this kind of technology in the nanopharmacology research, especially in the TCM studies, we can use this kind of approach either way, evaluating the therapeutic efficacy or investigating the mode of action, or we can starting either with the herbal interactions. You probably can record. I just mentioned one of the case in the is the 君臣佐死, the comparative rules in Chinese. And of course, we can use this kind of technology to dissection the active ingredients from the the traditional Chinese medicine, which is very complex, composed by hundreds of thousands of compounds. So for the network construction, I believe the AI-based approach will play a very important role in the near future. And of course, I believe that if we use this kind of new technology, especially in my slides, I just mentioned the single cell technology, and the spatially resolved transcriptomics, and the cell-cell communication will greatly enhance the role of a transcriptomics-based nanopharmacology, which I personally believe is very important for the TCM modernization. Okay, next slide.
Okay. 55.
Okay, so is the last one. Before I conclude, I'd like to acknowledge the incredible team efforts behind my presentation here today. The special thanks to my collaborators, both at my Zhejiang University, and also my colleagues at the US FDA and several others area. And also I'd like to appreciate the IUPHAR for the invitation for the Pharmacology Education Initiative. Is a very great honor for me. And of course, I'd like to truly appreciate the teamwork from my lab. All the glories actually belongs to the students and the staffs in my labs. So with that, I just stop it here. And again, I'm very sorry for the internet problem here. I'm very sorry for that. That's the first time I ever met. And I appreciate your patience and understand. So probably I'll stop it here.
Yep. Thank you, Dr. Fan. Thank you so much for the great presentation. And we understand technical problems happen, and we learn from them as we go. And I think in the future, it would probably be a good idea to ask for slides in advance so that we can immediately take over and present the slides when this happens. And internet connections also go bad. So we saw that in the beginning, and we saw the problems you were having, and that's absolutely nothing you could have done to change or control that. We're now going to move over to the Q&A portion, and I am also going to share a slide first, there's a slide for all the attendees so that you can claim your certificate of attendance using this code in the link or the QR code. And while this is up on the screen, we're going to start the Q&A. So if there are any questions, you can place that into the Q&A. The speakers or the microphones and the cameras are not active for attendees, but we will read the questions for you for Dr. Fan to respond to. Are there any questions?
Okay, Dr. Fan, this is Li Qin here. Probably I'll be the first to raise questions.
Yeah,
well,
thanks.
well, someone others probably is still trying to raise their questions. So, well, first of all, it's really amazing that to see all those great technologies that you can. Well, it's the amazing part is that like TCM is a very complicated system, and well, you kind of use those like I mean all those sequencing and all those methodologies to unify it or somehow to make it more readable, and that's what I think the amazing part. Well, I guess my question is, since like every person is different, if you do the whole genomic sequencing or transcriptome sequencing, the baseline it could be very different, right? So, there any way to offset that, or are you gonna standardize it, or what are you gonna do? Because like the personal difference are could be something to affect the results, right?
Yeah, absolutely. Yeah, first of all, thanks, Li Qin. Thanks for the great question. Actually, that's one of the key problem in terms of technology or the reproducibility in this area. There's a lot of discussions how many samples should we use in terms of the get reliable or reproducible results. So, so back to your question. Of course, there's a lot of difference in terms of the person, or even in terms of the different cells, as I mentioned, right? That's why we developed not the way a lot of people they develop this kind of a single cell omics approach. So there are always the question is how many samples should we use at the end of the day, but unfortunately so far we don't have the exact numbers for the animal study. In the most case, right, we use even like the numbers the animal is like n equal to like 5 to 10 or something. But is that enough or not? Honestly, we have no idea, right? For the clinical study, it's very important to estimate the sample size and the numbers. So of course, right now we do have a bunch of approach. So the algorithm in terms of data science, we can deal with this kind of a problem and decrease this kind of difference between the samples using this kind of algorithm. One of the common they use the approach with several genes we call housekeeping genes. They basically they should express the probably pretty much like the same across the samples. We use this kind of approaches. Of course, there are several others, but at the end of the day, we still have we don't have the kind of consensus regarding your questions.
Okay. Well, seems like Dr. Fan is still talking. Can you hear me?
Yes.
Oh, great. Probably
Thank you.
was my. Oh, great. Yeah, good to know. Good to know. Well, I think for any new technologies, it could be there's still questions to deal with, but but I'm still hoping to see more great advancement there from your group.
Thank
I
you.
guess there's a question from the audience. So how transcriptomic analysis require validation using wet lab studies? So do you do wet lab studies to validate?
Yeah, of course. Yes. That's actually, I believe a lot of researchers, especially students, I probably quite interesting about that. The first answer is sure, of course. The validation should be performed, especially if you need to publish your papers. For instance, you submit a manuscript to our pharmacology research. I believe that the first stage, the editor will ask for that. So there's several ways or several degrees of the validation in terms of validation. The first, you need to validate the data you got is actually is correct, right? So you basically you need some like the RT-PCR to validate the gene expression data you measured from the RNA sequence is actually the right. And the second level of the validation is your biological findings, right? Is meaningful. So you probably need to verify your findings through another set of cell research or the animal models. So as I introduced in most of the case I briefly introduced today, yeah, all followed by validations especially at the animal model.
There was a follow-up question to that same question from a Dr. Shah, asking, can you suggest any methodology steps for the same?
Excuse me, I didn't really follow the question.
It was following the question about the wet lab studies, asking, can you suggest any methodology steps for this?
Oh, okay. Yeah, I just mentioned several methodologies. The first, in terms of validate the data you obtained from either with sequencing or microarray or this kind of the single cell omics data, you need verified or validated the accuracy of the data. In most of the case, we're using the RT-PCR. I perform the same samples to make sure the data is correct. Okay, although we're using a different technology. Another, of course, you verify, for instance, like we identified a group of the compounds, they are active ingredients. So you needed to validate this kind of findings using a different approach. Of course, there are a lot of approaches here. I cannot specify them, but there, of course, there are a lot. We can, find the exactly methods through the papers.
Okay. Another question has come in from an attendee named Alam, and the question is, how can transcriptomics-based network pharmacology contribute to the personalization of TCM therapies?
Yeah, that's absolutely the great question. Honestly, I have to say, so far we still have in the ongoing research in this kind of area. We don't have the exact answer about that. One of the reasons is in most transcriptomics studies, as I mentioned, we're using this kind of bulk sequence approach. So we can only get the gene expressions, the upgrade or downgrade. So of course, this kind of data is actually very is patient by patient, right? So if one of the resolving approach is just as you mentioned, if we're using this kind of patient-divided samples, is of course a very important area. Although in my lab, we also do some organized research, which I believe is also belongs in the patient-divided samples. But so far, we in the TCM research area, it's still just at the very beginning. So we still don't have the consensus here in this kind of research area, and sorry for that.
Professor Cabrini asked first, she said she congratulates you on your excellent work and the excellent presentation, thanking you for it. She said there's no doubt that transcriptomic analyses have improved our understanding of herbs and TCM as well. Thank you for your incredible contribution. Based on your experience, do you consider that the use of herbs continues to be preferable to isolated compounds? Or considering the synergism of compounds, would it be more important to maintain the complex use of herbs?
Yeah, actually, first of all, thanks, especially in this kind of, awkward situations. I appreciate your patience. And regarding your question and suggestions, of course, an investigation is kind of isolated ingredients or compounds, of course, it is very important. Here in China, we also have a lot of researchers working in that area. And however, in the traditional Chinese medicine, we're still using this kind of prescriptions. I mean, in the clinical, they are not using the single compound. They're using this complex or the combination of the herbs. So that's why we still working a lot, put a lot of efforts in this kind of herbs or group of the herbs. So that's why because we try to understand why this kind of Chinese medicine works in the clinical side. Of course, we also investigate the single compound, isolated compounds, but that's again leads to another way, leads to probably we can pure compounds to at the end of the day we can come up with a new drug, but that's not the traditional Chinese medicine.
We have another question from Alam asking, how do transcriptomic responses to TCM differ among various cell types or patient-derived samples, for example, in cancer versus autoimmune disease?
Oh yeah, I believe I just answered some of this question. Yeah.
Okay. Then I will go to the next one. This is from Professor Joka. If transcriptomics and proteomics data are combined in an integrated analysis showing overlapping pathways regulated by the drug, to what extent is validation in the lab necessary?
Yeah, that's great. Actually, first of all, if you find the overlapping pathway, congratulations, right? That's actually a lot of researchers struggling for that. It's not easy to do combine the different omics data together in the same research in the same area. So regarding your questions, I believe if you find there's the overlap pathway, which actually will provide more confidence of your biological findings. And of course, in terms of addition, we can perform in the different ways, right? The most important thing in the pathway, we can interfere this kind of pathway to see what happens after, if this kind of pathway is really truly important, you should see some, response in terms of the model or the drug's treatment.
Okay. Hervey Shaw is asking, are transcriptomics, genomic, and proteomics can be correlated, combined in natural products research?
Yeah, of course. Although in the slides I mentioned here introduced here today, they mainly focus on the transcriptomics. But even in my lab, we also did a lot of proteomics and metabolomics. We try to integrate this kind of data together to answer our questions. But as I also mentioned, it's not that easy. Although we can perform the experiments, so we got the data, but at the end of the day, it's pretty hard and pretty challenging to integrate all these kind of omics state together. You probably find, in some case, even very contrary, totally different, the findings in terms of the, different omics.
Yeah. A question coming from Luis asking, first says, thank you for the insightful presentation. Then asking, since cells are the key mediators of therapeutic effects, more so than individual genes, how does transcriptomics-based network pharmacology address the complexity of cell-cell communication? Specifically, how can we define and differentiate a balanced versus a disrupted cell-cell network in the context of TCM treatment? Furthermore, given that cell-cell communication often occurs across different organs, can current approaches truly capture a holistic, system-wide view, or are they still limited to single tissue perspectives?
Yeah, I have to say I very much enjoyed this kind of insightful question. Actually, this is also a lot of discussion in my lab in terms of pretty much in the same questions. We're still working on this kind of area. First, I'd like to say our role as kind of a cell-cell communication or cell-cell communication network is of course different, right, between the samples, right? Since we're using this kind of, I just mentioned, we can inference this kind of network, cell-cell communication network from the single cell data or spatial transformation data. In most of the case, we actually ignored this kind of I had to say the difference between the individual samples or the patients. So you probably in some cases you probably will get the different cell network from the different patients or different samples or different animal models. That's very common. So one of the resolving approach for this kind of situation, you probably can find from several manuscripts and several published papers, as they pull all the samples together and sequence them and using this kind of data to inference the cell communication. That's also we in our lab we also adopt this kind of approach to decrease this kind of the effects the sample in terms of the different samples. Another interesting the questions that you mentioned about is for the cell communication actually often occurs in different organs, right? Yeah, that's actually a good question actually. Unfortunately, so far, especially the single cell omics, we mainly perform in the same organ. So that's one of the reasons. One of the major problem right now, we don't have the enough data, to inference the cross-organ cell-cell communication. That's probably the problem here, and not even mentioned, not only the different organs, right? We also need to investigate, the such as sorry, I just saw that here. Yeah.
Okay. A question coming from Alam. What challenges arise in validating transcriptomics-based network models in vivo, especially when studying multi-compound herbal formulas?
Basically, for the in vivo, I mean, it's actually generally is okay. I think in our lab we actually using this kind of technology and for over 10 years, and we from my point of view and our personal experience, I think it's in most of the cases, it works very well. And so I don't think there's a problem in terms of validating this kind of models in vivo. However, you mentioned about the multi-component urban formulas, since it's quite complicated in terms of the TCM or the urban formula. It's in most of the cases there are hundreds of thousands of compounds, and the quality control of this kind of formula is extremely important in this kind of research. You need to make sure, the samples. I mean the urban samples, their quantities, the batch-to-batch consistency, in most cases are controllable. That's one of the things you need to consider at the very beginning.
Okay, there's 2 more questions, and then I think we should cut off the questions there as we've gone a little bit over, but we also had technical problems, so we went a little bit late. This one coming from Purvi Shah. I'm very much fascinated with the work from your research group. I want to learn this in depth and explore for traditional knowledge. I have a background from natural products chemistry. Can you suggest to me what do I start with? How do I start?
Okay, I assume he's a grad student. In that case, if you are interested about this area, first I'd like to suggest you guys and read some papers in this area, realize the scenario or the technology behind this kind of technology. And as for the second, in most of the cases, if you want to use this kind of a transcriptomics-based approach, you need to deal with a lot of data. So you probably need to learn some, bioinformatics or the chemometrics approach. So in that case, you probably need to learn some, the modeling language such as Python or R, so that you can, perform, as I mentioned, several algorithm or R package to deal with your own data. So that's my 2 cents.
Okay, and the last question coming from, it looks like the name is Mo Junyu. How do you efficiently and simultaneously determine the contents of various active ingredients in traditional Chinese medicine TCM?
Okay, it's actually right now we already have a bunch of technology to can efficiently resolve this kind of problem. Especially we can use in the mass spectrum to identify the ingredients and quantitatively determine this kind of contents. So that's not a difficult problem anymore. Of course, back to like 20 years ago, it's very difficult. But right now, I don't think there will be a problem as long as you have these kind of equipments. Or you can send out your samples to some experiment lab as long as they have this kind of mass spectrum.
Okay, thank you so much, Dr. Fan. I'm going to stop with the questions there. One more came in, but if you have further questions, please feel free to write to us at the email address that's shown on the screen, and we will forward the questions to Dr. Fan for him to respond to, if that's okay with you, Dr. Fan.
Yeah, no problem.
Okay. Well, thank you very much. Thank you for all the attendees, and apologies again for technical issues. We will be posting the recording online within a few days. And thank you so much, Dr. Fan, for your wonderful presentation.
Thanks for having me. Of course, I apologize for the technical problem.
That's no problem at all. These things happen. Okay.
Bye.
Thank you. Bye.
Thank you. Thank you so much.